Hi there, my name is Ana Jimenez. I am at Colgate University. Today I'm going to be talking to you about metabolic consequences of aging in small and large breed dogs. And my thanks goes out to Dr. Campanella for inviting me to be a part of this summit. And so before we get going on the research that my lab has been doing, I want to talk to you about the theory behind these experiments. And so when we look at lifespan across different body sizes, across different species of mammals, what we know is that there's a positive correlation between body mass and longevity in between species, mammalian species, whether they're in the wild or whether they're in captivity. However, if we look at dogs, a single species, you will be able to see that this relationship actually is inverted, so that a small body size will yield a long lifespan compared to a large body size. And to say that in a shorter way, a Chihuahua will easily live 16 years, whereas a Great Dane is fairly old at 9 or 10 years old. And so I'm a comparative physiologist, and this relationship between body mass and mean lifespan was really interesting to me. I should say that there are other mammalian species that do demonstrate this lifespan, horses and rabbits, but I am interested in the dog model. And so these data were first published in 2016, and what that led me to do was ask several questions at the cell level. And so these are data that I'm presenting to you at the whole organismal level, but at the cell level, my lab started looking at two different questions. First, there's cellular metabolism, metabolomics, and oxidative stress differences between small breeds and large breeds. And secondly, are there ways to manipulate the cell architecture or the cell metabolism to affect lifespan in dogs? And so we're going to take these questions in turn. I'm going to address the first question, and then I'm going to address the second question. And so you'll notice that there's actually quite a lot on the first question. And so I'll walk you through each of the things that I have on here. We're going to start with cellular metabolism, then we're going to talk about metabolomics, and then we'll talk about oxidative stress. Within these different aspects of metabolism, we're going to talk about different tissue types. And so I will alert you when we're switching tissue. And so let's look at this first question first and foremost. Why do I care about cellular metabolism? Well, in 2013, there was a paper that came out that highlighted the hallmarks of aging. And two of the hallmarks of aging actually deal with the mitochondria. This is the organelle that's responsible for nutrient sensing and overall ATP formation. And so the two hallmarks of aging that are associated with this are, as I said, nutrient sensing and mitochondrial dysfunction. And so if there are hallmarks of agings that are dictated by this organelle, there may be some pretty drastic differences in the rate that this organelle works in small breed and large breed dogs. And so to address this question, we used a cell type called a primary fibroblast cell. And so some people may be familiar with the cell types, others may not be. A primary fibroblast is a cell type that is found in all tissues of the body. They are mainly responsible for connective tissue formation and for wound repair, right? And so if you look at this picture right here, this is what they look like underneath a microscope. And this picture right here is a confocal image. The red that you see there are mitotracker stained mitochondria. And so you can see, and the blue is a nuclei. And so what you can see is that these cells do have mitochondria. And so why do we use primary fibroblast cells to address metabolism questions? In tissue culture, the relationship between the nucleus and the mitochondria are kept even when those cells are isolated from the organism. So that is to say that, for example, this nucleus, if I isolated it from a large breed dog, it will have the phenotypic metabolic signature of that large breed dog. If I isolated it from a small breed dog, it'll have the phenotypic metabolic differences of a small breed dog. And so this is sort of a really good tissue type to look at these sorts of questions. To first start addressing the cellular metabolic differences between small breed and large breed dogs, we had to isolate a whole bunch of these primary fibroblast cells. And so we have a total of 4 groups that we look at. We have large breed dogs, we have puppies of large breed dogs, and we have adult dogs of large breed dogs that we isolate primary fibroblasts from. We also have small breeds, puppies and adults. Now, these tissue or these primary fibroblast cells were isolated from routine surgeries at the puppy level and from older euthanized adults. And so the particular primary fibroblasts that we use are dermal fibroblasts. And so we need about a thumbnail size piece of skin to be able to grow these tissues. And so once we had a bunch of primary fibroblast cell lines from each of these 4 categories of dogs, what we did was start to measure their cellular metabolic rate. And so I'm going to talk to you through some methods before I show you some results. And so we have these primary fibroblasts. I like to remind you that what we're trying to figure out is why there is that negative relationship between body mass and lifespan, mean lifespan, in small breed and large breed dogs that's opposing to other mammals. And so if there are whole animal differences, there should be cell level differences as well. And so to do these experiments, we use a machine called a seahorse. It is not, in fact, a seahorse, the animal. The machine was called a seahorse because of the 2 owners of the company. What this machine does is we have a 96-well plate here, and we plate our primary fibroblasts in duplicate in these wells. What the machine does is it lowers a micro cartridge, right? So that there is a very, very, very small chamber that's formed. There's mitochondria within these cells, and those mitochondria are consuming oxygen, right? And so for a short period of time, this cartridge lowers, mitochondria are consuming oxygen, then you get a measurement like this, where the slope of that line, the cartridge is raised so that the cells can then get oxygen, and the slope of that line ends up being the rate of oxygen consumption. Now you will see that there are 2 lines on here. The seahorse is a really great machine because it can, at the same time, in real life, measure oxygen consumption rate, and so that's aerobic profiles. And it can also measure anaerobic rates. And so this is this ECAR. Anaerobic rates refer to the rates of glycolysis. ECAR stands for extracellular acidification rate. And so we can measure both rates of metabolism, the aerobic and the anaerobic. I'm first going to walk you through the aerobic experiments and results, then I'll walk you through the anaerobic experiments and results. So if you look at what the seahorse is now owned by a company called Agilent. And so if we look at the methods that Agilent provides to measure the typical oxidative metabolic profile, these experiments look like this. And so I'm focusing on here. You'll see that the first 3 measurements are going to be our basal oxygen consumption. And so that is the oxygen consumption of the cells when nothing else is present, right? Then we can add a drug called oligomycin. Oligomycin decreases oxygen consumption so that we can measure ATP production and proton leak. And proton leak has been linked to lifespan. Oligomycin binds to the F1-FO ATPase. And so it decouples ATP production. Then we can inject the cells with a drug called FCCP. FCCP is a drug that is like a shot of adrenaline to the mitochondria. And so it gives us a maximal respiration rate of those cells. We get some measurements after the FCCP, and then we inject 2 other drugs. Antimyosin A binds to complex 3, and brotenone binds to complex 1. That shuts down all mitochondrial respiration, and it gives us a proxy of non-mitochondrial respiration. There are other things in the cell that do consume oxygen, and so that's what that measurement is for. And so now that I've walked you through these typical oxidative profiles, I'm going to show you some results for the first time. I want to first guide you to how the next couple of graphs are oriented. What you see here are the puppies, the primary fibroblast rates of puppies. This is, of small dogs. This is a small puppy. This is a small old dog. This is a large breed puppy. This is a large breed old dog. And the next couple of graphs are going to be like that. And so, again, I want to remind you that I'm looking for where are there metabolic differences that could explain that whole animal metabolic rate of the inverse, large breed dogs living long lives and small breed dogs living long lives and large breed dogs living short lives. And so our results from about 221 dogs in this data set showed that basal oxygen consumption rate, that is just without any drugs whatsoever, demonstrated a higher rate in older dogs, despite the size class, right? And then we also see that there's an increase in proton leak in older dogs as well, despite of size class. And so what this implies is that regardless of size class, small or large breed dogs, there's going to be an increase in inefficiencies. And so we see that by an increase in basal OCAR and the increase in proton leak. This is not new or special. We know that as organisms age, their metabolic rate becomes more inefficient. And so there is nothing in the aerobic profile that explains that lifespan to body mass relationship between small breeds and large breed dogs. If we move on to the anaerobic profile, this is again what Agilent, how they describe their anaerobic or glycolytic profile. And so these are all processes that are happening within the cytoplasm of these cells. And so I'll guide you through these again. We have our cells in the 96-well plate, and the seahorse is measuring this. The first 3 points are again going to be basal glycolysis. Then we inject, the first 3 points are non-glycolytic acidification. We inject glucose, and then the process of glycolysis can start. And so we get basal glucose after the basal glycolysis, after the glucose injection. We inject oligomyosin, and that increases rates of glycolysis to give us a glycolytic capacity. And then we can inject a compound called 2DG to shut down all glycolysis and bring us down to our non-glycolytic acidification. Now, I'm going to show you graphs. These graphs are set up exactly the same way as the previous 2. Small breed dogs here, large breed dogs here. I got it right that time. And so when we saw these results, this is when things became very interesting to us. It is unfortunate that it's not good news for large breed dogs, or at least so far we don't think that it's good news. And so let me walk you through this. These are all the 3 processes that I just walked you through in the previous graph. What you see here is that regardless of the glycolytic process, you have large breed dogs having higher rates of glycolysis, significantly so, compared with small breed dogs. And so why is this? So this is interesting to us, but it's not good news for large breed dogs. And so why is that? An increase in glycolysis can mean two things. An increase in glycolysis can mean an increase in lactate formation, or it can be an increase in glucose oxidation. The increase in lactate formation is actually really, really bad news. That's called the Warburg effect. And it's a process that is linked to tumorigenesis. And so it's a process that's linked to cancer formation. As most of you may know, a lot of large breed dogs often die of cancer. And so the increases in glycolysis, which are linked to increases in insulin growth factor 1, that's something that's in the literature, both of those things make it so that large breed dogs are in a tumor-like phenotype for all of their lives, right? Puppies as well as large old dogs have an increase in glycolysis. And so this is not great news. To figure out where in the glycolytic process this was happening, we needed a method that was significantly more detailed than what we could do with the seahorse. And so this is where now we're going to be moving into metabolomics. We're done talking about cellular metabolic rates for now. We are still talking about primary fibroblast cells. And so I'm going to show you a graph, and you're going to see lots of things, but I'm going to walk you through it. And so this is work that I did with Paul Brooks at the University of Rochester Medical Center. And this has already been published 2 years ago. And so what metabolomics allows us to do is to determine concentrations of all different kinds of molecules. And so you can see here, all of this, you don't have to know all of this is the process of glycolysis. And all of these molecules, we can determine the concentrations of them. And so here you have small breed dogs here, large breed dogs here. Again, you have puppies or young dogs and old dogs here. And so two things that we need to figure out, or that we need to explore here. Are we getting increases in lactate formation? And so that would mean it's the Warburg effect. The Warburg effect is a precancerous phenotype, right? Or is it glucose oxidation? And so if we move all the way down here, you'll see that this is lactate right here. And what you can see is that there are no increases of lactate in the large breed dogs, and there's no significant differences in lactate concentration in any of our four groups. And so it's thankfully not the Warburg effect. And so what is it? Is it increases in glucose oxidation? We don't have evidence that that's it either. What we do know, if you look here, here's our four groups again. What you can see here is that in the lower half of glycolysis, here, likely, there is a deficiency, a metabolic deficiency in large breed dogs that is not present in small breed dogs. And so we're not out of the woods yet trying to figure out whether this links to cancer formation or not. We are currently exploring whether there are any associations between this pathway or this pattern and the P53 signaling pathway and the SIRT1 signaling pathway. And so we haven't finished here. What we do know is that it is not the Warburg effect, which is great news, but that there is a metabolic deficiency in the lower half of glycolysis for large breed dogs. Okay, so now we're going to move away from primary fibroblast cells, and we're going to move into a different tissue to answer a different question. And so the tissue that I'm using to answer the next question, or the next part of the first question, is going to be blood. And what we're doing is we're looking at the concept of oxidative stress. And so why did we look at this? The early why do large dogs die young literature blamed it on oxidative stress, where there were no data that explained why large breed dogs would have an association with oxidative stress. And so we sought to figure out whether this was something that was a culprit for large breed dogs dying young. And so first, let me tell you what oxidative stress is. And I'll tell you to start off that oxidative stress is a byproduct of aerobic metabolism. So like 6 slides ago. So let me walk you through this. All mitochondria of both of these examples are going to take in oxygen, and they're going to, at the exchange of oxygen, make ATP. Oxygen, O2, is a paired electron molecule, but that O2 molecule in the process of metabolism can split, leaving those two O2 molecules with an unpaired electron. That unpaired electron has a high electronegativity and really, really, really wants to find another unpaired electron. There are unpaired electrons everywhere in the cell. There's unpaired electrons in lipids. There's unpaired electrons in DNA. There's unpaired electrons in proteins. If this oxygen molecule splits and there's an unpaired electron, this unpaired electron is going to bounce around in the cell, lipids, DNA, proteins, and cause damage, right? And so that's what we call oxidative damage. That's right here, right? Luckily for most organisms, we have a developed system within our cells to deal with these pro-oxidants. And so some people in the literature call them reactive oxygen species, ROS, or reactive species, RS. And so each cell has a developed antioxidant capacity system. This antioxidant capacity system can be endogenous. It can be what our cells are making, endogenous, or it can be exogenous. It can be coming from our diet, right? And so this is why blueberries, for example, are so good for us, because they have a whole lot of antioxidants. And so if we have a system, 2 mitochondria, that are making ROS at the same rate, but that have differences in the antioxidant capacity, that could mean differences in the amount of oxidative damage those cells accrue over time. And so if you have a poorly developed antioxidant capacity, you're going to have a lot of damage. If you have a well-developed antioxidant capacity system, you're going to have very little damage. What we were interested in for this question was not so much the exogenous or diet side of antioxidant capacity, but the endogenous or the cell level side of antioxidant capacity. So there are 3 enzymes that we care about a lot. And so those are superoxide dismutase, I'll call this SOD, catalase, I'll call this CAT, or glutathione peroxidase, and I'll call this GPX. These 3 enzymes serve to thwart any unpaired electron damage within the cell. And so usually in the literature, you will see these 3 enzymes called the cellular armament of the cell. And so anytime that anyone says cellular armament, this is what I picture. It's a little army truck. And so in what we know from the whole organism, what would we expect to see? And so we know that small breed dogs live long lives compared with large breed dogs. And so 2 things can be happening here. Either, or two things isolated from each other, or 2 things together. Either small breed dogs have a smaller rate of reactive oxygen species, and or they have a significantly well-developed antioxidant system, which then means that they could have a decrease in oxidative damage, which then could lead to shorter lives because of less oxidative damage. And then in turn, large breed dogs could have an increase in reactive oxygen species and or a less developed antioxidant capacity system, which then together or separately could yield to them having higher oxidative damage. The link with oxidative damage is that usually when you have oxidative damage, you start accruing errors, which lead to disease. And disease can also be cancer here. And so let me walk you through what we did. And so remember at the beginning of this, I said that we were going to use blood for this question. And we use blood because blood is a metabolic reservoir for this particular process. And so that is a good tissue type to figure out the whole organism what's happening with oxidative stress. I will also tell you that the data that I'm about to show you are not just on domestic dogs. Excuse me. We wanted to compare domestic dogs to wild canids. And so wild canids are anything from wolves to dholes to coyotes, that kind of animal. And we wanted to do that because if, for example, you're comparing a wolf to a similar sized dog, what you'll see is that the wolf, being the same size, will live significantly longer lives than the same size dog. And so we wanted to see whether the wild canids could tell us something about what's happening with domestic dogs. For this paper that I'm about to show you, it was published in 2020, and we phylogenetically corrected not just the wild canids, but also the domestic dogs. And so all of the data that I'm about to show you have phylogenetic corrections within them. And so what are the things that we measured? Measuring reactive oxygen species, because they're ephemeral, is very difficult. And so what we measured was the components of antioxidant capacity in the form of TAC, that stands for total antioxidant capacity. It's the total pool of antioxidant molecules that will help thwart the reactive oxygen species. Then we measure the activities and concentrations of the three enzymes that I mentioned, CAT, GPX, and SOD. And then furthermore, we measured lipid oxidative damage. When we're talking about oxidative stress, you can't talk about oxidative stress by just measuring the antioxidant pool or just measuring the lipid damage. It's a seesaw. And so you have to measure both. You have to measure the antioxidant capacity and some type of damage. And so let me show you some of these results. I'm going to guide you. This is a lot. I'm going to guide you through what these graphs stand for. So first, you'll see that this is lipid damage. This is total antioxidant capacity. This is again total antioxidant capacity. And this is GPX activity. What you'll see in the white or colorless dots, those are our domestic dogs. And the relationship that we're looking at in this panel is age at which the blood draw occurred. What you're seeing here in C and D is a corrected measurement of age. When we say, here's a 2-year-old Chihuahua, it is not the same thing as saying, here's a 2-year-old Great Dane. And so to try to correct for the discrepancies in aging rates, what we did was we corrected the age at which the blood draw occurred to the, it's called maximum lifespan. And we obtain mean breed lifespans from the American Kennel Club page. And so let me show you some of the results. What you see in colored dots are going to be all of our different wild canids. And so you'll see we have Maine wolves. We got all of these from zoos, Arctic fox, coyotes, dholes, so forth and so on. What you see here is that there is a significant correlation between lipid damage and age, uncorrected age, in domestic dogs. So the older a domestic dog gets, the higher the oxidative, the lipid oxidative damage is. And so this is what we would expect with the theory of free radical theory of aging, right? But then we found something really interesting in the wild canines, which is that instead of increasing damage, the wild canines actually increase most aspects of their antioxidant capacity. And so instead of accruing damage, they're actually trying to do better to thwart that damage. And so what you see here is an increase in total antioxidant capacity in wild canines. And then when we correct for age, you still get an increase in total antioxidant capacity and an increase in GPX activity only in the wild canines. And so domestic dogs have an increase in damage. Wild canines have an increase in everything that's good for them to thwart that oxidative damage. And so that's one thing that they have in common. If we look at body mass, what you'll see is this. Again, the domestic dogs in this gray line demonstrate a decrease in lipid damage with increasing body size. And so that's not what we expected. If you think back to a couple of slides ago, what I told you was that we expected large breed dogs to have an increase in lipid damage, an increase in oxidative damage that would be linked to disease, that would be linked to the short lives. And what we find is the exact opposite. What we find is that small breed dogs have a significantly higher lipid oxidative damage concentration compared with medium-sized dogs and compared with large breed dogs. And so this is in complete opposition of what most of the literature says, and in complete opposition to most of the comparative literature as well. We do know that there are organisms that have higher lipid damage across mammals, but they're sort of the weirdos in the world. And so things like bats have higher lipid damage and can live long lives. And so this is still something that we're working out. We don't know whether the higher lipid damage in small breed dogs comes from a higher reactive oxygen species formation, which could be linked to their higher mass specific metabolic rates, or whether it's a clearing. So it's something related to kidney function, that they have a slower rate of clearing throughout them. And then what we see again with the wild canines is that they actually, as their body size increases, they invest in that antioxidant capacity. And so there are some links between the longer lifespans of wild canines that are employing a strategy of building antioxidant capacities in comparison to domestic dogs who are just letting damage build up. And so again, this is a continuing question that we are trying to sort of tease out. Okay. And so now I know that that was a lot for the first question, but we did cover cellular metabolic rates. And remember, we talked about glycolysis being the weird thing that we found in large breed dogs. And we measured oxidative stress, and oxidative stress, we found pretty much everything that is opposing to everything in the literature, with small breed dogs having higher rates of damage. What we're going to move on to now is asking the question of, are there ways to manipulate cellular architecture to alter cellular metabolism in dogs that can affect lifespan? And then I'm also going to present you a very short snippet of a paper we did on commercially available drugs that may or may not extend lifespan. Just check how I'm doing with it. I have 6 minutes. I can do this. I'd like to acknowledge the following people who have helped me with this project. Josh Winmore was a student in my lab for about 4 years, and then Alex Champagne and Kenneth Walsh, who were at the University of Southern Indiana. They measured all the lipid data that I'm about to present to you. And again, this paper was published in 2020. Here's the theory behind this. And so all these graphs that I'm showing you are from Tony Holbert's work. And what I want to show you is how we came to design this question the way that we designed it. First, I know I'm showing you mammals and birds and we're talking about dogs, but remember, I'm a comparative physiologist. So when we look at mammals and birds, mammals in white, birds in dark, what you see is that for similar body sizes, birds live significantly longer lives compared with mammals. Tony Holbert's work tried to figure out why this was. And so what he figured out is looking at skeletal muscle phospholipids and at liver mitochondrial phospholipids was that the shorter your life, the higher your peroxidation index for both skeletal muscle and mitochondrial and liver mitochondrial phospholipids. What that means is that the lower the peroxidation index, the longer you're able to live. And so remember, we're still talking about small breed dogs having long lives and large breed dogs having short lives. And so if we dissect out what that peroxidation index means, what we're doing is we're going from cells down to cell components, and we're looking at the lipid architecture within those cells. And so if we look at the lipid architecture within those cells, the thing that increases peroxidation index are double bonds in membranes, right? And so these are called polyunsaturated fatty acids, right? You have that double bond. And the more double bonds you have, the higher your peroxidation index. And so the more double bonds you have, you should have a shorter lifespan, essentially. And so we sought to design an experiment to test this out. Luckily for us, we had already had some basal information on oxidative stress. And so the concept itself, this is called the membrane as a pacemaker theory of aging. The concept of it, the concept itself is this: lower membrane polyunsaturation, a decrease in number of double bonds, means that you have a lower mass specific metabolic rate, lower mitochondrial oxygen consumption, lower formation of reactive oxygen species, and overall lower oxidation stress. Higher membrane polyunsaturation, higher number of double bonds means the opposite. You would have a higher mitochondrial oxygen consumption, higher reactive oxygen species production, and higher rates of oxidative stress. And so how can we test this? Well, we went back to fibroblasts. I will tell you two things that you need to know here. We only use puppy primary fibroblasts, and we treated each of we isolated cells from one individual, for example, and we treated those cells, we divided them and treated them in control ways, and then we exposed them to a high concentration of oleic acid. Oleic acid is a monounsaturated fatty acid, and so that would decrease the total hypothetically decrease the total peroxidation index of these cells. Luckily, primary fibroblasts are sort of the ideal tissue to do this in, because they will soak up and incorporate the free fatty acids floating in the media that we're growing them in. And so the first thing we needed to figure out was whether in fact these primary fibroblasts were changing their phospholipid architecture, right? And so this is an example of all the different phospholipids in one set of cells. The DMEM are our control values, and our OA are our oleic acid values. And what you can see is that oleic acid is 18.1, right? And so you can see that oleic acid here has been very much taken up by the cells. And not only is that particular fatty acid been changed the structure of those cells, but also this particular fatty acid change. This is called DHA, and DHA has been a phospholipid that's been linked to lifespan. And so it was really interesting to see that in one individual, just for exposing the cells to oleic acid, the DHA concentration also changed. And so once we knew that we were in fact changing the structure, the architecture of the cell, in these primary fibroblasts, what we did was we went ahead and measured aerobic metabolism that we had done before. And so if you remember to 2 slides ago, if we decreased the peroxidation index, we would increase mitochondrial respiration. And that's in fact exactly what we found. Here we have small breed dogs, the control and the oleic acid, and large breed dogs. Remember, these are just puppies. And what you see is that there is a significant decrease in maximal respiration. And so that's check mark. Decreasing peroxidation index does decrease maximal respiration, which could be linked to increases in lifespan. But everything else we found was not good. And here's what I mean by that. First and foremost, I want to talk about spare respiratory capacity. Spare respiratory capacity is a component of cellular metabolism, which is the amount of buffering energy that cells have. And so the larger the spare respiratory capacity, the more flexibility that cell has to deal with something in an emergency state, right? And so here's what I want to show you. There's actually, with oleic acid treatment, there's a decrease in spare respiratory capacity. So if these cells had any type of an energetic emergency, they would not be able to respond to it. So this is not good. Oleic acid actually does the opposite of what we want. And then what you see here is that in large breed dogs, there is no difference with oleic acid or without oleic acid in spare respiratory capacity. This is a caution that we cannot have the exact same treatments for small breed dogs and large breed dogs. What this tells us is that they have different energetic requirements and they don't respond similarly to a similar treatment. Then what we have here is coupling efficiency. This is coupling efficiency refers to the amount of oxygen coming into the cell that gets turned into ATP. And so coupling efficiency is also kind of bad news bears, because the coupling efficiency for both large breed dogs and small breed dogs decreases. And so we are getting less ATP from each oxygen molecule. And so again, this is just a sort of a cautionary tale that like we did do the thing that would increase lifespan, but it has some fairly negative metabolic consequences that I would not want to see at the whole animal level. Now, we are building on these results, and we're using other fatty acids currently to try to figure out whether we can develop media that are ideal for each different size of dog. And so stay tuned for that too. As I said before, I want to very quickly walk you through cellular metabolic rates of primary fibroblasts in the presence of 3 different commercially available life-expanding drugs, and that's metformin, rapamycin, resveratrol. And so what you have here is the result of a glycolytic profile, and what you have here are the results of an oxidative profile. These are small puppy, small old, large puppy, large old, right? And so what we found was when we exposed these primary fibroblasts to these different drugs, the only treatment, the only drug that actually demonstrated life-expanding, lifespan increasing metabolic signatures was rapamycin, which you can see here. You see a decrease, an increase of basal oxygen concentration. And this life-expanding metabolic rate only happens when the dogs are treated as puppies. We sort of lose that ability to change the cell metabolism in large breed or old dogs. Metformin and resveratrol actually did things that oppose the changes of cellular metabolic rates that we would expect with increases in lifespan. And so again, just a cautionary tale that Not all commercially available expanding drugs do similar things in different types of dogs. And so with that, I have a bunch of people to thank. We would not be able to do the work we do without the support of a great veterinarian staff, and also the support of many, many breeders. Paul Brooks at the University of Rochester has been a huge help to our lab, and Colgate University has uniquely funded these projects. And so I usually get asked why I ended up in the dog aging world, and she's the reason why. Her name was Emma. And with that, I would love to take some questions.